You know, when we talk about saving our planet's wildlife, the sheer scale of the challenge, well, it can feel overwhelming sometimes. We're facing really widespread biodiversity loss, habitat degradation just continuing, and of course, climate change accelerating things. And for generations, the way we've monitored species ecosystems, it's often felt like we're constantly playing catch up. It's just been incredibly labor intensive, right?
Hmm.
Often needing us to physically interact with, maybe even disturb the animals we're trying so hard to protect. But what if there were, say, two revolutionary tools emerging that are really starting to flip this script? Today, we're taking a deep dive into environmental DNA, that's eDNA and bioinformatics. And together, these technologies, they aren't just small improvements. They seem to offer genuinely groundbreaking, scalable solutions for conservation.
Absolutely. And our mission for you, the listener, today is really to explore how eDNA lets us detect species with, well, unprecedented precision and with minimal disturbance, which is key. Then we'll look at how bioinformatics comes in to make sense of just the massive amounts of genetic data that gets collected. The real goal here is to show you how these tools are empowering smarter, faster decisions, in this urgent race to save wildlife.
Okay, so let's unpack this then, starting with eDNA. What exactly is it?
Right. So think of it as the genetic traces organisms leave behind. Naturally. Could be skin cells, maybe hair, waste products, bits of themselves shed into the environment.
Ah, okay, so it's like genetic breadcrumbs.
Exactly. And this DNA, it disperses into the surroundings. Could be water, could be soil, sometimes even the air.
And that is where it gets really powerful, I suppose. The ability to detect, well, a whole range of species, even the really elusive ones or endangered nocturnal animals,
Hmm.
without ever needing to actually capture them or even see them.
Precisely. That's the non-invasive breakthrough.
So you could, I don't know, confirm a rare amphibian is in a pond just by testing the water, without netting or anything.
That's exactly it. You collect environmental samples, water, soil, snow.
Yeah.
Maybe not the animal itself. It's a total game changer for sensitive ecosystems and, well, shy creatures. And the applications are pretty broad. For instance, monitoring endangered species. And lets conservationists confirm if a threatened species is present without stressing it out. We've seen it used really effectively for, say, amphibians or freshwater fish in these super fragile ecosystems where traditional surveys might just cause too much disruption.
Mm-hmm. Minimizing disturbance.
Yeah. And it's also a fantastic early warning system for invasive species.
Mm.
If you could detect them rapidly using eDNA, you can intervene much quicker before they, wreak havoc ecologically.
So finding that invasive carp DNA downstream before you actually see the carp. Yeah.
Exactly. It buys crucial time. And then there's assessing overall biodiversity. eDNA surveys can give you this comprehensive snapshot. Who's living in this ecosystem? It's
Hmm.
incredibly useful in places that are hard to reach or just incredibly diverse, like rainforests or coral reefs. Trying to survey those traditionally is a nightmare.
Right.
We can even potentially track migrations. By taking samples over time, temporal sampling, we can get clues about animal movement, which is vital for conserving species that move around a lot, like whales or migratory birds.
Okay, so you collect these samples, water, soil, whatever it is. You've got this invisible library of genetic material. But then how do you actually read that library? You must end up with just vast amounts of raw genetic data. How do you translate that into, species X is here?
Right. And that is a huge challenge. You've got this microscopic goldmine, like you said,
Yeah.
but it's raw data. And that's exactly where bioinformatics comes into play.
The computational side of things.
Exactly. Bioinformatics is essentially the set of computational methods, the tools, algorithms designed specifically to organize all that data, analyze it, and ultimately interpret it.
Hmm.
Without bioinformatics, you just have terabytes of, well, essentially genetic noise. It wouldn't tell you much directly.
So what does bioinformatics do with that eDNA data in a conservation context?
Well, several key things.
Yeah.
First, species identification. This is fundamental. Bioinformatics tools match the DNA sequences you found in your sample against huge established reference databases. Think of libraries like GenBank or BOL, the Barcode of Life data system. These databases contain known DNA sequences for millions of species. So the software finds a match, and boom, you know what species that DNA likely came from.
Okay, so it's like a genetic fingerprint matching system.
Pretty much, yeah. Then you have things like phylogenetic analysis. This helps us understand the evolutionary relationships between the different species detected. How closely related are they? which could be really important for conservation priorities, maybe focusing on genetically unique lineages, for example.
Ah, okay.
Not just what's there, but how it fits into the bigger tree of life.
Precisely. And we can also look within a species using population genetics. Bioinformatics tools analyze genetic variation within that species' DNA from the sample. This gives us crucial insights into things like inbreeding risk, gene flow between populations, overall genetic health. It's like a checkup for the population's gene pool.
That sounds incredibly detailed.
It can be. And finally, you can integrate this genetic data with other information, ecological data, climate data, geographic data, using bioinformatics for habitat suitability modeling. This helps predict where species might thrive or where they might struggle, especially with things like climate change. It helps guide where to focus conservation efforts strategically.
Right. So eDNA finds the clues, bioinformatics deciphers them, basically.
That's a good way to put it.
So when you bring these two together, eDNA providing the raw material and bioinformatics providing the interpretation,
Yeah.
what's the combined impact? Is it really, transforming things on the ground? Oh, absolutely. The synergy is where the real power lies. It enables rapid and crucially scalable monitoring. Think about traditional methods, camera traps, physically surveying areas, catching and tagging animals. They're vital, but often very labor-intensive, expensive, and limited in how much ground you can cover or how often.
You can't have traps everywhere all the time.
Exactly. But with eDNA, you can potentially sample much larger areas more frequently, maybe monitor an entire river system or large forest plots. And bioinformatics ensures that the flood of data coming from those samples gets processed relatively quickly. This allows for much more timely decision-making.
So faster insights across bigger scales.
Yes.
Well,
And that leads directly to more evidence-based conservation. When you can precisely identify which species are present, track changes in their distribution or abundance over time, conservationists can make decisions based on solid, current data. Things like, where should we prioritize protecting habitat?
Yeah.
Are protected areas actually working for the species we think they are? How do we need to adapt our management strategies?
so it makes conservation less reactive, perhaps more proactive.
That's the goal, definitely. And there's another really interesting angle here: citizen science and policy integration. We're seeing the development of more portable eDNA sampling kits, and bioinformatics platforms are becoming, well, a bit more user-friendly, more accessible.
So non-specialists could potentially get involved.
Potentially, yes. It's empowering local communities, citizen scientists, to actively participate in monitoring their own environments, which is fantastic for engagement and data collection. And the data generated, whether by researchers or citizens, can then be fed into larger national or international biodiversity databases. This can directly inform environmental policy and even help countries meet their commitments under things like the Convention on Biological Diversity.
That's fascinating. Democratizing data collection almost.
In a way, yeah. It broadens the scope significantly.
Now, this all sounds incredibly promising, almost like a silver bullet. But are there challenges, limitations we need to be aware of? I mean, it can't be perfect.
No, definitely not perfect.
Right.
And it's crucial we acknowledge the limitations. Critical thinking is absolutely essential. One major challenge is data gaps in those reference libraries we talked about.
Right. If a species' DNA isn't in the library,
You might misidentify it, or worse, just miss it entirely. It comes up as unknown. So expanding and curating those reference databases is a constant, ongoing effort globally.
okay, that makes sense.
And then there's the contamination risk you alluded to earlier. eDNA is incredibly sensitive. A tiny amount of DNA from somewhere else, from the researcher, from equipment, from another sample, can potentially lead to a false positive.
So finding DNA for a species that isn't actually there.
Exactly. So really strict, meticulous protocols are absolutely vital during sample collection in the field, and then again during processing in the lab. It requires extreme care.
I can imagine. Handling tiny invisible traces.
Yeah. And finally, there are complexities in interpretation. Just detecting DNA doesn't automatically mean you have a living, breathing, viable population right there, right now. DNA can persist in the environment for a while after an animal has left or even died. It can be transported by water or wind.
Ah, so finding fish DNA doesn't mean the fish is swimming there today.
Not necessarily.
Yeah.
So bioinformatics analyses need to consider these factors: the timing, the location, the environmental conditions, to accurately interpret what the presence or absence or abundance of eDNA really signifies about the actual organisms and their populations. It's not always straightforward.
Okay, so context is everything, even with this high-tech approach.
Always.
So, wrapping things up a bit, it seems clear that the integration, this combination of eDNA and bioinformatics, it really is reshaping wildlife conservation. It's giving us tools for these incredibly precise, fast, and non-invasive ways to assess biodiversity, almost like giving ecosystems a regular health check.
That's a great analogy. And yes, these tools are genuinely helping scientists, managers, policymakers make much more informed, data-driven decisions, ultimately to better protect ecosystems and all the species living within them. And the future outlook is exciting too, as the technologies continue to improve, get cheaper, more portable, and as data sharing gets better globally, their role is only going to grow.
It really feels like a significant leap forward, so something for you, our listener, to maybe ponder. As eDNA and bioinformatics do become even more widespread, more accessible around the globe for conservation, what sort of new ethical questions might pop up, or what new opportunities for global collaboration could emerge from this shared technology?